Good morning or afternoon. Today what I'm going to do, we did our first lecture, or Monday's people, you'll get yours Monday. This is kind of the second bit of the lecture, so I'm going to have to give you a little bit on video because we only need an hour and 15 minutes. We're supposed to have 2.5. So I'll try and put about 20 minutes up. It's about as big a video as I like to upload. So 20-minute one, then maybe one more 20-minute one before your next lecture. So this is kind of lecture 2. You have the intro, we started off. So what I'm going to do is go over a little bit more kind of the general characteristics of the endocrine system. Again, this should be chapter 17 in your textbook. And again, there's a Learn Smart posted already over chapter 17. Start working on that. It'll make you kind of force you to hit the textbook a bit. Let's see, we looked at, last time we were comparing kind of the nervous system, endocrine system. Endocrine system is relatively slow, nervous system is fast. Endocrine system is sustained, nervous system has got a pretty short duration. Nervous system is pretty specific, only a few cells are affected by motor neuron. Endocrine system, while it does vary, it can have very broad effect, like thyroxine, for example, affects just about every cell in the body. Things like ADH, a little bit more specific, but still a broader range than a nervous system. The next thing we'll look at has to do with sort of the way you control the reaction to stimulus intensity. Nervous system, we mentioned, is frequency controlled. Kind of how fast the nervous system generates action potential is going to affect the response of the effector, the muscle or gland or whatnot. In the endocrine system, it's more or less a dosage response or amplitude for the response. In general, the higher the dose, the more response from your target cell. So that's the general rule. There are some weird exceptions though, because of the way the system works. It can be very hard to predict what a particular dose of hormone will do at any given time. Let me give you an example. So I'm going to scribble on the whiteboard here. I'll overlay at the top videos, a little clip from some of the PowerPoints if I need a diagram. Mostly I'll be scribbling stick pictures. Sorry about that. All right, so let me do a diagram, something like this. Let's say this y-axis will represent the response of the target organ. Just relative scale. This is a strong or high response. This is pretty low. Over here, x-axis, basically is just going to be time. Now here's what we're going to do. I'm going to take a hormone. Doesn't matter what it is. Let's call it hormone X, because it doesn't matter what it is. And at this point in time, right here, I'll call it T equals 0. I'm going to take a dose of this hormone. What's the dose going to be? Let's say 10 micrograms per liter. This is a small dose. I inject this 10 micrograms per liter into the subject and I get a response. Let's just say, for the sake of argument, this is the response. That's how much the permeability changed. That's how much protein synthesis you have. Whatever the response for that hormone is for the target cell, this is it. That's kind of good. Now all I'm going to do, I'm going to take the same subject, same hormone, same dose, but now I'm going to move over a little later. Let's say this is T equals 24 hours. So it's the same individual, same drug. I'm going to apply the same dose, 10 micrograms per liter. Apply the same dose to the same individual, and then I look at the response, and let's say, for the sake of argument, I get something like this. Same hormone, same individual, same dose, but the effect is much more, it's much greater. There's more permeability or more protein synthesis, something. How is that possible? All right, what can happen in some cases? Remember, when we talk about target organs, target cells, there is a specificity. That is, a target organ or target tissue or cell has to have protein receptors for that particular hormone. The number of receptors a cell has is not necessarily fixed. You can make more of a protein, less of a protein, it depends. So just to give you an idea, let's say I'll make a little red diagram. This represents the target cell for hormone X. And let's say hormone X, I'm going to make it like a little red triangle. Of course, that hormone is released into the blood, diffuses the tissues, diffuses out of the blood, And this target cell, if it's a target cell, it must have receptors for that particular hormone if it's going to respond. So the hormone is released, it comes in, and it binds to the receptors, and I get this response. Now, 24 hours later, if we look at the same target cell, let's say it looks something like this. Those are supposed to be receptors, by the way. They're not little hearts or anything, but you know, those years of art school didn't pay off. So these are the receptors. Again, the same dose of hormone is released, but now because you have more receptors, whereas over here all the receptors were full, so there's some hormone just sitting there, can't do anything. Now you have more receptors, so the response is greater. The idea is the number of receptors for a particular hormone is not fixed. Sometimes, just the effect of the hormone binding to that receptor can stimulate the cell to start producing more receptors. So it becomes more sensitive, or some other environmental factor can cause a production of more receptors. But in this case, this may be called up. They call it upregulation. All it means is the response of a target cell to a particular dose of hormone will go up over time. Even though it's the same hormone, same dose, the target cells are more sensitive, but they respond more. That's upregulation. Now, we could also do, if I can find an eraser. There we go, got one. I think this is, I think I was the last person to use this room like in March. Let's do a similar thing, except, same scenario. I can change the hormone. Let's make it hormone Y now. Again, it doesn't really matter. I'm just changing its name for the heck of it. But we give this hormone Y to the subject here. You get this response, you know, that same dose, 10 micrograms per liter or something like that, just a typical dose of hormone. Everything's fine. But now we come back 24 hours later. I apply the same dose of the hormone, but let's say now I get a response that looks something like this. Same hormone, same dose, same subject. Response is reduced. It's not nearly as strong. What could happen? Again, imagine here's your original target cell. It's the same sort of thing, except this time, if this is the target cell, the number of receptors can be reduced. So now when the hormone comes in, it doesn't have as many places to bind. You get a reduction in response. 3 guesses what they call that. It's often referred to as down regulation. That is, the response of the target cell to a particular dose of hormone can be blunted over time. You know, what can happen is one of the effects of the hormone binding to receptors of the target cell is it can cause a target cell to degrade more of the proteins. So there's fewer receptors available, the response will be blunted. You know, you get to see something similar with what is it, the type 2 diabetes, where the hormone insulin is still present, but when it's released, it doesn't affect the target tissues because they've basically been overstimulated. It's a reduction in a response of the target system, blunted response, blunted sensitivity, so your blood sugar levels are controlled as they should be. That is one thing to keep in. That's why it's kind of hard to figure out, you know, therapeutically hormone doses. They can vary a bit over time and within an individual, because, you know, normally you increase the dosage, you increase the response, but occasionally you get, you know, kind of a backwards response that you wouldn't expect. It's upregulation, downregulation, response can vary over time. So that's something to keep in mind. But again, as a general rule, more hormone, more of a response, with a few exceptions. All right, that's upregulation, downregulation. Oh, the next thing we'll do, this is fun. Chemical characteristics. Now, there are people who make a living, you know, on the chemistry of hormones. Chemistry is very complex, of course. Every hormone has to be distinct chemically, so it can respond or bind to one particular receptor. We're not getting into any of the details of the structure. It's beyond the scope of this class, and it's beyond the scope of the instructor at this point. But, you know, you can take hormone chemistry courses. There's a lot to it. What we're going to do, we're going to keep the chemistry a little simpler. We're going to look at just kind of some chemical classes of hormones. There's really just 3. And realistically, functionally, I'm just going to separate them into 2 functional chemical categories. So it's not going to be too bad. Let me do the first bit. All right. chemical types of hormones. Usually your textbook will split them into 3 different types. You have peptide hormones. You may remember from last semester, you messed around with peptides a bit. Peptide hormones are basically strings of amino acids. They're proteins or fragments of proteins. These are the most common types of hormones. Most of the ones we're talking about in class will be peptide hormones. It's the bulk of them. There is another category called the monoamines. The name's pretty self-explanatory. They are made out of one modified amino acid. Some examples would be epinephrine is a monoamine, thyroxine is a monoamine, it's a modified amino acid. So there's a handful of monoamines. And then the last class, or last group, will be the steroid hormones. You looked at steroids last semester based on cholesterol. These are lipid-based hormones. They are made from modifying a molecule of cholesterol, which you might have looked at last semester as it makes a component of our cell membranes, for example. Cholesterol is a steroid. It's used as a precursor for many of the steroid hormones. So there are 3 chemical classes, and I think in that hormone table in your text, they'll discuss or describe whether it's a peptide hormone, monoamine, or steroid. What I want to do, because functionally, to make it even simpler, there's really just 2 sort of chemical functional groups of hormones. This will determine how they interact with the target cell. So I'm going to actually reduce this to 2 sort of functional groups. The first one, non-lipid soluble. Non-lipid soluble hormones are typically the peptide hormones. Those big proteins don't get through phospholipid bilayers. Again, this is going to be the importance. We're talking about lipid solubility. Remember, cell membranes are basically a layer of lipid. If you're not lipid soluble, you cannot easily cross phospholipid bilayers, which means it's hard to get in and out of cells. And of course, the blood vessels are also lined with layers of epithelia. Essentially, that's a layer of phospholipid bilayers, several of them. So non-lipid soluble hormones are not going to get in and out of cells easily or in and out of the blood easily. Most of the non-lipid soluble are peptide hormones. Most of the monoamines are also non-lipid soluble. I'll mention more about these in a sec. Over here, we'll go to the steroid hormones. Steroid hormones are, of course, lipid soluble. They're made of lipids, cholesterol based. Now, the only exception, all of your steroid hormones are lipid soluble, and one of the monoamines is lipid soluble. So all the monoamines are non-lipid soluble except thyroxine. It's the exception. So the only lipid soluble hormones are your steroids and thyroxine. And here's a way to remember them. You can go through there's a table in the book, and I think the one I gave you, it'll tell you whether it's lipid soluble, non-lipid soluble. You can memorize them all, don't bother. What you're gonna have to eventually is determine, what hormones come from what gland, right? Like the posterior pituitary secretes ADH oxytocin. The testes, testosterone, the ovaries, estrogen. So you're gonna have to know what hormones come from each gland anyway. So here's a way to remember if they're lipid soluble or not. The steroid hormones only come from 2 places. If the hormone comes from the gonads, ovaries or testes, it is a steroid hormone, it's lipid soluble. That's estrogen, progesterone, and testosterone. If the hormone comes from the adrenal cortex, which we looked at briefly in lab, or we will look at in lab, the hormones from the adrenal cortex, which are examples I usually use, are aldosterone and cortisol, and of course testosterone as well, or other androgens. Those are steroid hormones and lipid soluble. So basically, if it comes from the gonads and it comes from the adrenal cortex, it's a steroid, it's lipid soluble. And you'll have to know those anyway, so that's it. The only exception is thyroxine, which is not a steroid, is lipid soluble. So if it's not thyroxine, it doesn't come from the gonads, and it doesn't come from the adrenal cortex, it's non-lipid soluble. And that functionally is probably more important than knowing whether it's a monoamine or a peptide hormone. Is it lipid soluble? Is it not? Because as we'll see next, that's going to determine how the hormone's going to behave, how it's going to interact with the target cells, how it's going to move in and out of the blood. So that's the last thing I'll do in this video. We'll look at a quick kind of description of how the lipid-soluble hormones work versus how the non-lipid soluble hormones work. Let's do lipid-soluble first, because they're actually a little simpler, I think. Now, I'll draw an ugly picture on the board, and I'll go ahead and put above us somewhere the diagram that's from your PowerPoint, because it's a little prettier than mine. Let me switch to it here. All right. So, lipid-soluble hormones. Which will equal the steroids plus thyroxine. Again, it comes from the gonads, the adrenal cortex, or it's thyroxine. Those are all lipid-soluble hormones. Let me turn on this recorder here real quick. All right. Sorry about that. Get the video up. I put it in the top screen here. It is off the PowerPoint. It won't be as clear on the video, so you can refer to the PowerPoint if you need to. All right, lipid-soluble hormones, the way they work. First of all, let me put a gland here. And I'll put a blood vessel here. And I'll put a target cell over here. As with just about all the hormones, the hormone is going to be produced. I'll make the hormone a little black thing here, a little black triangle. It's released from the endocrine gland into the interstitial fluid out here somewhere. And as we saw in our definition, the hormone then has to make its way into the blood. So this hormone, at a high concentration here, wants to diffuse into here. Again, blood vessels are lined with epithelial cells. They call it endothelium. So it's basically just like a sheet of phospholipid bilayer. It's cell membrane. In order to get across from outside the blood vessel here to inside, you're going to have to be able to move across this phospholipid bilayer. Well, of course, the steroid hormones are lipid soluble. So basically, it's like the blood vessel isn't there. It's just going to diffuse right in, no problem at all. Once it gets into the blood, of course, it gets distributed throughout the body. And again, when you reach your target cell, here are all the hormones in the blood. They're at a higher concentration than they are out here. These hormones will freely diffuse out because nothing's going to stop them. They're lipid soluble. They move right through the phospholipid bilayer. Then, these hormones, I'll draw it again as a little triangle, when they reach the target cell is surrounded by a phospholipid bilayer. But again, if you're lipid soluble, it's like it's not there. This hormone will readily diffuse right through the phospholipid bilayer and get into the cell cytoplasm. Now here's an important bit. The lipid-soluble hormones, they diffuse directly into the cell and they bind to receptors that are either in the cell cytoplasm or more commonly in the nucleus of the cell. Let's say here's your nucleus here. Nucleus with the DNA, all the genes, that sort of thing. So lipid-soluble hormones, they readily enter the cell. They'll diffuse into the cytoplasm, into the nucleus of the cell, and what these do is the hormone binds to a receptor inside the cell, and typically what all the steroid hormones and thyroxine do, they're all lipid-soluble, they bind to a receptor in the nucleus that's usually associated with the genetic material. What these lipid-soluble hormones will typically do is cause new gene expression is often the way you'll see it worded. Basically, they go into the nucleus, bind a receptor, which causes a portion of the DNA, a gene, to be expressed. That means it gets read, transcribed and translated into a new protein. So typically with these lipid-soluble hormones, you're going to end up producing a new protein from scratch. This is typically why, when we look at the speed that some of these hormones work, like I think I gave the example of thyroxine takes like 10 days to see the maximum effect. This is partly why all the lipid-soluble hormones, they come into the target cell and they cause new proteins to be produced. That's a slow process. So things like thyroxine, there's a 10-day lag. Testosterone, puberty doesn't happen overnight. It's a pretty slow hormone, and that's typical for all of the lipid-soluble hormones because they have to cause new proteins to be produced from scratch. Again, with thyroxine, remember it ups metabolism? Well, metabolism is due to those metabolic pathways you looked at last semester. Glycolysis, Krebs cycle, electron transfer chain. What are those metabolic pathways? There are a bunch of enzymes, a bunch of proteins. So one of the ways thyroxine will up your metabolism, it basically causes you to produce more mitochondrial enzymes. So you can break down glucose faster, make more ATP, and so forth. So typical for lipid-soluble hormones, they cause new proteins to be produced, and they will be slower hormones. So it's kind of the direct mode of action. These lipid-soluble genes go into the DNA, cause new proteins to be produced. Now let's look at the non-lipid soluble. They'll be a bit different. Let's start another view here. All right. Sorry, getting the other camera set up. Let me look at the non-lipid soluble hormones. I'll redraw my same diagram, except we're looking at non-lipid soluble hormones. Again, these are all peptide hormones, all monoamines, except thyroxine. All right, so I'll have the same sort of scenario set up. Here's the gland, here's the blood, here is the hormone. So the gland will secrete the hormones, like before, previous example, into the interstitial space. It's going to diffuse into the blood. However, these are non-lipid soluble hormones. This blood vessel is lined basically with phospholipid bilayer, all those epithelial cells. They can't get across. Non-lipid soluble proteins don't move across phospholipid bilayers worth a darn. So what's going to happen? The hormone would never get in the blood, you'd be screwed. It's kind of useless. So what happens, sort of developmentally, the blood supply for endocrine glands that secrete non-lipid soluble hormones has to be modified. These blood vessels typically will have a very high permeability. They'll describe them as being fenestrated capillaries. They have little slits open, there are pores in them. Basically, the epithelial cells may not line up all the way. There's a crack in them. This is an avenue that allows the hormones to diffuse into the blood. Again, there's small openings. They don't allow blood cells to leak out or anything else. But you have to modify the permeability of the blood vessels to allow these non-lipid soluble hormones in. So they make it into the blood, okay, just like before. But now we have a similar problem. Here's a target cell. Let me make this one bigger. Now, when the hormone reaches a target cell, you've got the same problem. It's at a high concentration in the blood. How's it going to get out here? It can't move across the phospholipid bilayer. So again, the blood supply of the target cells has to be modified a little bit. It's going to have a higher permeability, fenestrations, pores, what have you. That will allow the hormones to diffuse out of the blood. But now we have another problem. When the hormone reaches a target cell, it can't get in. It's not lipid soluble. So to never get in, what's it going to do? Well, on the target cells for non-lipid soluble hormones, the receptors are not in the cytoplasm or in the nucleus. They are membrane receptors. You know, they're like transmembrane or integral membrane proteins you looked at last semester. They're sitting in the membrane, but they're facing the outside. So now the hormones don't have to get through the phospholipid bilayer. They just come in, they bind to these receptors, and that's going to cause a response. Now, the response is going to be a little different. That's what we'll look at next. These non-lipid soluble hormones, they don't have a direct mode of action. They don't cause new genes to be transcribed, translated, new proteins to be formed, nothing like that. They use what's called a second messenger system. Now, there are several second messenger systems that are used. If you look in your textbook, I think they'll give you at least 3. They give you something called an IP3 second messenger system, calcium calmodulin second messenger system. So there are others out there. I'm only going to look at one. It's probably the most common, and we understand it pretty well, so that's one I'll use as an example. The little video that'll be up here somewhere or over here, that is a diagram showing the cyclic AMP second messenger system. So that's what we'll look at. So non-lipid soluble hormones, they use it's called a second messenger system because the hormone is the first messenger, but as we'll see, a different signal or a second messenger will be produced inside of the cell as a response to that hormone binding to the receptor. So we're going to look specifically at cyclic AMP second messenger system. That's cyclic adenosine monophosphate. Again, you usually see the abbreviation. So cyclic AMP. And to try and describe it sort of briefly, I'm going to make myself a big target cell here. My drawing will be a little ugly, but again, you look up top, you'll see the publisher's version of it. It's a little prettier. But let's say this is a target cell. All right, we'll have the receptors in the membrane, receptor, and the hormone will come in and bind to the receptor. Now that's all the hormone's ever going to do. It binds to the receptor, it won't do anything else. But remember, this receptor is a protein. When things bind to it, that can often cause a conformation change or a shape change. It can change its activity. That acts like a signal. So when the hormone binds to this receptor, what it's going to do is it's going to sort of send a signal, usually via various conformation changes, to another protein that is referred to as a G protein. Not very creative with the naming, but it's called a G protein. It's a pretty common signaling protein. It's also in the membrane. I think it's a peripheral protein. Now what that G protein is going to do, it's going to send a signal to another membrane protein. I'll just kind of draw it as a big blue potato here. This membrane protein is called adenylate cyclase. It's a membrane protein, but remember, one of the functions of membrane proteins, they can have enzymatic capability. This is an enzyme. Adenylate cyclase is normally an inactive enzyme. That is, it doesn't normally do anything. But when it receives a signal from the G protein, that activates or sort of turns on the adenylate cyclase. Now, what does adenylate cyclase do? Adenylate cyclase is an enzyme that will take ATP, remember that one, adenosine triphosphate, common in the cytoplasm. That's what all the metabolic pathways produce. Living cells will have ATP in them. And it then enzymatically converts it into cyclic, I'll just put a C down there, cyclic AMP. That is the second messenger. Normally, if that hormone wasn't present, that cyclic AMP wouldn't be in the cell. It's not there at all. But as soon as that hormone binds to the receptor, it basically causes a production of this cyclic AMP. That's the signal. Now, what does it do? I'm going to give you a big enzymatic cascade. The diagram will be pretty to see because I may skip a few steps. But basically, what cyclic AMP will do is it takes inactive protein kinases. These are basically enzymes that will act on proteins within the cell, and it turns them into an active form. Now, what do the active kinases do? The active kinases will then take other inactive enzymes. You see, you have a boatload of enzymes in the cell. They're already pre-made. They're just in a configuration or conformation that doesn't allow them to have enzymatic activity. These protein kinases will take the inactive cell enzymes and convert them into active enzymes. And you looked at enzymes a little bit last semester, but they catalyze chemical reactions, speed them up, allow them to occur at lower temperatures. So what's this active enzyme going to do? These active enzymes will take whatever the substrate is. Again, a little waffly here. Substrate is just what the enzyme will work on. It could be a variety of things. It depends on the hormone that we're talking about in the target cell. Substrate, and turns it into products. So again, look up here or over here, and that'll give you a prettier diagram. But that's the idea. So this is referred to as the cyclic AMP second messenger system. This is the way, the second messenger system is the way the non-lipid soluble hormones will work. The cyclic AMP second messenger system is the most common, but there are others. Again, IP3 is a modified phospholipid, basically. That's used as a signal. Calcium, calmodulin, calcium is a common signal as a second messenger as well. But if I want any details, it'll be this. So you should probably know the steps. I think the diagram up there kind of has them listed, step 1, 2, 3, 4, and it's covered in your textbook as well. Looks a little pretty in this. I'm going to quit there because that's been a little over, been close to 30 minutes, and that's about all I can upload. So I will see you in a bit. Bye.